Hello everybody and welcome to this biology video on enzymes and factors affecting them. So the first thing that you need to understand and get into your head before I begin any of this is that enzymes are proteins. So they are going to have a primary structure, a secondary structure and tertiary structure. They're going to be made of amino acids. They're going to have bonds between those amino acids, between the amino acids R groups. So enzymes are proteins, and that's what you need to understand for this whole part. So firstly, how does heat affect enzymes and their rate of reaction? Well, firstly, if I increase heat by a small amount, that's going to give the substrate and the enzyme more kinetic energy. Therefore, they're going to be able to move around a lot faster, meaning the substrate and the enzyme are more likely to collide. Therefore, meaning that more enzyme substrate complexes will form, therefore I will have an increased rate of reaction. Catalysis will happen faster. However, if I increase the temperature too much, then we get this process of enzyme denaturing happening. And this is kind of, again in a bit more detail, step up from what you learned at GCSE. So if I increase the temperature too much, the amino acids, individual amino acids within the protein, will begin to vibrate. And due to that, the weak bonds in the enzyme structure, in the protein structure, will begin to break down. I've written there breaks hydrogen bonds in the primary structure. That doesn't make sense. What will happen is hydrogen bonds and ionic bonds in the secondary and tertiary structures will be broken by these amino acids vibrating, and this will cause the enzyme to unfold. It will lose its tertiary structure, it will lose its 3D shape, so the enzyme will just kind of flop, it will lose its structure completely. And due to this, the active site shape is going to change, and that means that the enzyme can't catalyse the reaction anymore because the substrate won't fit into its active site. So therefore the enzyme has denatured and it can't work at all. And this tends to be permanent once you heat it up too high. If I cool it down again, the enzyme won't sort of snap back into the shape that it should be as the bonds reform. That doesn't tend to happen. So if I increase temperature a little bit, more enzyme substrate collisions, more enzyme substrate complexes, increased rate of reaction. But if I increase it too much, then the amino acids will vibrate as they get given more kinetic energy. Therefore the hydrogen and ionic bonds in the enzyme secondary and tertiary structure will break. The enzyme will unfold, loses its 3D shape, the active site will then change shape, substrate can't fit into it, therefore the enzyme is denatured, can't catalyse the reaction. Next we have how pH will affect enzymes. So firstly, what do we mean by pH? If we have a low pH, that means we have acidic conditions, but acidity is caused by hydrogen plus ions. So if we have a low pH, we have lots of hydrogen plus ions, and these hydrogen plus ions will either be attracted to and bond to by an ionic bond or a hydrogen bond, R groups on the outside of the enzyme, and that will have a knock-on effect on all the other bonds within the enzyme, causing the tertiary structure to alter, active site changes shape. That means it can't catalyse the reaction anymore because the substrate won't fit into it. Or the hydrogen plus ions could just generally interfere with the hydrogen and ionic bonds, again altering the tertiary structure, 3D shape changes, active site shape changes, therefore the substrate can't fit into the active site shape anymore, therefore the enzyme can't catalyse the reaction. That's relatively simple. That happens if we have a low pH. But also if we have a low pH, lots of hydrogen plus ions, if we have any negatively charged R groups in the active site, on the active site itself, then these positively charged hydrogen ions are going to be attracted to those R groups. So therefore the active site is going to be clogged up by all these hydrogen ions, meaning the substrate can't fit into it anymore. The temporary bonds in the induced fit hypotheses won't form. Therefore the enzyme can't catalyse the reaction, so therefore catalysis doesn't occur, quite simply. So the enzyme's optimum pH that we speak about is the one which gives it the best active site shape. So the one where there will be X amount of hydrogen ions and they won't clog up the active site too much, they won't interfere with the bonds, the ionic and hydrogen bonds in the tertiary structure, and they won't form new bonds on the outside of the enzyme. That's the optimum pH, when we have just that right amount so that no negative impacts happen on the enzyme. So its tertiary structure doesn't change shape, so the active site remains its proper shape so it can catalyse the reaction with the substrate being able to fit into it properly.